Testing. Okay, good morning everyone. Welcome to lecture 25 of PMS 300. We're almost halfway there. Right, so today we're going to just wrap up a few things from the nervous system which I did not get to yesterday, and the majority of today's class will be focusing on the muscular system, which is the next organ system which we'll be talking about. So this is the slide where we stopped at yesterday. We were talking about the differences between the sympathetic and the parasympathetic divisions of the autonomic nervous system. And I was talking about this last point. Most vital organs receive dual innervation, right? Which means that, for example, if you take the heart, notice that it's got innervation from the from the sympathetic division as shown right here, and also it's got innervation from the parasympathetic division as well. And this is very important because it helps to maintain what is known as an autonomic tone. Right, and autonomic tone is where the motor neurons, they're going to maintain a level of background activity even without any stimulation. Right, so it's maintaining a certain level of background activity without any stimulation. And when you think about why this is the case, it's very important for having a regulatory effect. So when you think about regulation, I want you to think about maybe the speed of a car. Right, if a car was stopped and it was if it was turned off, all you can do is turn it on. But then if the car is moving, then you have a chance of modulating the speed. And so similarly, this autonomic tone and the background level of activities like the moving car, and so you can have better control over. You want the heart rate maybe to go up or to go down in the case of the heart. So an organ like the heart, which has dual innervation, there will be both the release of acetylcholine, which is the neurotransmitter released to target organs when you have the activation of the parasympathetic division, and there will also be norepinephrine, which are the hormones released in response to sympathetic activation. So both these will be released continuously to maintain this autonomic tone. And when you're at rest, we will have the parasympathetic division is going to dominate under resting conditions. But whenever there is a time of stress, or there is a crisis, we see that stimulation for the sympathetic innervation is going to happen, and the parasympathetic innervation is going to be inhibited. All right, so it's like the parasympathetic division will be inhibited and the sympathetic will be activated during a time of crisis, and that's what will lead to an increased heart rate. And we will see how there will be a change in heart rate, when we talk about the cardiovascular system, we'll see how the breathing rate is modulated when we discuss the respiratory system. So those are details we'll consider when we talk about the respective organ systems and how the nervous system can control those organ systems. Are there any questions before we proceed? Okay, if there are no questions, now we want to see how the G protein coupled receptors, how they are going to be activated, and how there can be three different pathways which can take place. So here's a figure which you're all familiar with, which we considered in the endocrine system, and I told you that the hormone is going to be the first messenger, and this is a hormone which is hydrophilic, so it cannot get past the lipid bilayer, so it will bind to a G protein coupled receptor. And I said that once this hormone binds, this G protein here is going to be activated, and that activation will lead to an increase in the levels of cyclic AMP. So that's all the detail we considered until now. But what I want to tell you is that this G protein here is actually made up of three different subunits. There's an alpha subunit, a beta subunit, and a gamma subunit. And depending on the type of receptors, the alpha subunits themselves can be of different types. For example, we have a G alpha S subunit, can also have a G alpha I subunit, and in the next slide we'll see what is the relevance of a G alpha Q subunit as well. And so G-alpha-S and G-alpha-I both are capable of binding to the enzyme adenylate cyclase. Right? So adenylyl cyclase is the enzyme which will help in the conversion of ATP into cyclic AMP. And so if a specific receptor is bound, then G-alpha-S is released. In another case, G-alpha-I may be released depending on the different type of receptor. And G-alpha-S, the S subscript stands for stimulatory. What it does is it will help in the adenyl cyclase converting ATP into cyclic AMP. And so as a result, we see all of these downstream processes like other enzymes being activated, or maybe ion channels being opened, which is of relevance, especially in the working of the nervous system. We see all of this happens because of the G-alpha-S subunit having a downstream effect. Now, in case it was a different type of G protein coupled receptor and there is a release of the G-alpha-I subunit, what this does is it's going to bind to adenyl cyclase and it will inhibit it. And if adenyl cyclase is being inhibited, we see that the levels of cyclic AMP are going to decrease, and so you'll have the opposite effect, right? You won't have the various enzymes being activated further downstream. So this is just to show you that there are different pathways activated by the G protein coupled receptors, and we've talked about two of those in this slide. Does anyone need clarification on anything which was said until this point? So I've really simplified things, and I'm not telling you what are the different types of receptors because that is unnecessary details I don't want to burden you with. If you take a more advanced course, would you agree there's enough detail in the course as it is? All right, great. So let's talk about the third type of pathway, and this is a figure which you've seen previously where calcium is the secondary messenger. But in order for calcium to be released as a secondary messenger, we see that the G-alpha-Q subunit has been released, right? And what G-alpha-Q subunit does is it's going to bind to phospholipase C, and this will help in the conversion of phosphatidyl inositol bisphosphate, which is abbreviated as PIP2. It's going to be converted into diacyl glycerol and also inositol triphosphate. So the only detail I'm adding to this slide, which you've previously seen, is that the role of this alpha Q subunit, right? That's the only additional detail I'm adding. And inositol triphosphate is going to bind to specific receptors in the smooth endoplasmic reticulum, and as a result, calcium has been released into the cytosol, and calcium is required to activate protein kinase C, and the letter C is indicating that calcium is needed to activate it, but also diacyl glycerol, which was a product of the initial reaction, will also bind to protein kinase C. And protein kinase C will have further downstream effects which can play a role in smooth muscle contraction. Are there any questions on how the secondary messengers work? Yes?
So just the main factor for IP3 is that it just releases calcium in the cytoplasm?
Yes, as far as we are concerned. It's going to lead to the release of calcium from the smooth endoplasmic reticulum. Okay, if there are no other questions, that brings us to the conclusion of the nervous system. All right, so let me just summarize what we were discussing from yesterday. All right, so we were talking about presynaptic. What's funny, guys? What is that?
I just dropped something.
Oh, okay, so that's fine. I'm like, share the joke. There's enough tension in the room. All right, so we were talking about the presynaptic and postsynaptic regulation and how it can play a role in the generation or no action potentials being generated. So we talked about inhibitory postsynaptic potential, excitatory postsynaptic potentials, and we also saw how there can be regulation at the axo-axonic synapses as well. After that, we looked at the structure of the spinal cord, and one of the main takeaway messages from the structure of the spinal cord is how the input signals are going to be carried by sensory nerves through the posterior root to the spinal cord, and then output signals are going to be carried by motor nerves through the anterior root. So that was one of the main takeaway messages, and we considered that because that will help us to understand how reflexes work. So we talked about two types of spinal reflexes, monosynaptic and the polysynaptic reflexes, and I told you how polysynaptic reflexes will be much slower because you have more connections, and it's going to slow down the communication. After that, we talked about sensory pathways and how signals of touch can be carried all the way to the brain and how it's processed. So we looked at two pathways, the spinothalamic pathway and the PCML pathway. So the spinothalamic pathway will help to detect crude touch sensation, whereas the PCML pathway can help detect fine touch sensation, right? And the other difference is where the second order neurons, they will decussate, right? And in the case of spinothalamic pathway, they will be decussating in the spinal cord, whereas in the PCML pathway, they're going to decussate all the way in the medulla oblongata, which is part of the brainstem. And so because of the differences in where this decussation happens, in a condition like the Brown-Séquard syndrome, the affected side will lose the fine touch sensation, whereas the opposite side will lose the crude touch sensation. And also on the affected side, you will lose motor function as well. And so this is a concept which may be tricky, but you can expect a question on this. So if you need any additional help, please talk to us in the teaching team. And then we talked about the sensory and the motor homunculus as depicted here in these pictures, and this is just representing a map of how the brain areas are dedicated to processing sensory input and also motor output, and how different parts of the body will have different levels of dedication to these processing. And then we moved on to the differences between the parasympathetic and the sympathetic divisions of the autonomic nervous system, and I told you how there are different types of neurotransmitters released, right, at the target organs. In the case of sympathetic, you will have epinephrine and norepinephrine; those are the hormones released. Whereas in the case of parasympathetic, you have acetylcholine released to the target organs. And then finally, we wrapped up by talking about the three different pathways of G protein coupled receptors. When you have this alpha S subunit, you will have an increase in cyclic AMP levels, and then in alpha Q, it's going to increase the levels of calcium as a secondary messenger, and alpha I will decrease the levels of cyclic AMP. Okay. Any questions, clarification? All right, so the goal is to give all of you the review slides by Friday, right? By tomorrow, I'll give you the slides. If you do not go through the slides over the weekend and you just come and you look at it on Monday, it's not going to fulfill its purpose. So what I would say is, even if you have a very fun-packed weekend, just take 20 minutes, right, to look at the questions and see if you're able to answer it. And if you're not able to answer it, maybe you'll get motivated to start studying, all right? But if you do well, then maybe go ahead and, you don't have to be worried as much, right? But please go through the slides over the weekend. I know there's a whole mindset of I will not work on the weekend, but please, I'll give it to you on Friday, and you can go through those questions, and then when you come back on Monday, you can practice your understanding. What's your question?
Is there going to be a practice exam?
I'll have to check. Will there be a practice exam?
I wrote it. I sent it out for review.
Okay. Yes, there will be a practice exam. What was your question?
I was asking if we could get the practice exam by Friday.
Yes.
I already wrote it. I'm just waiting for it.
Just waiting for review. Yes, it's on my desk or my inbox. I will send it. Did you find the practice exam helpful?
Yes.
Yes. What about the review slides? Okay, good. We'll have both those ready for you by Friday so that you can practice over the weekend. All right, so exam 2, you will have up to this point, right? The nervous system won't be covered, sorry, the muscular system, whatever I'm talking about today and tomorrow, won't be on exam 2, all right? What was your question?
When is the key for the practice exam going to be?
Monday. Did you hear that? Yeah. Key for the practice exam will be released on Monday. So with that, we will now move on to the muscular system. Oh, you have a question here? Yes.
Is there going to be lecture objectives?
Say it again.
Is there going to be lecture